Okay, so excuse me. We are going to explore how to hand draw replication, but first we're going to begin with a basic review of the structure of DNA in a basic format. So here we see that we have DNA, and we can see here we have an individual nucleotide. So there's the phosphate group, the deoxyribose sugar, and a nitrogenous base, which could be A, T, C, or G because this is DNA. We can also see that we have the first carbon at that first vertex, the second, the third, the fourth, and the fifth, because deoxyribose is a 5-carbon sugar. We're ignoring all the hydrogens and oxygens except for the one key oxygen that's right there that's part of the pentagon shape of the deoxyribose, but we're ignoring the oxygens that are part of the hydroxyl groups. And we do see how the third carbon of the deoxyribose binds to the phosphate of the adjacent nucleotide. And so here now we see the second nucleotide of the same strand of DNA, and its third carbon binds to the phosphate of the next nucleotide right here. Okay, and so this is the 3' end, this is the 5' end, and we always say DNA is 5' to 3', and because we're working in an English language and English writing people established the convention, we always go 5' to 3' on the top of the DNA. Oh, let me resize this view. It's not quite the right size. There we go. That's a little bit better. Okay. And then this bottom strand, I know it got a little bit cramped, but we'll just deal with it. So we have a nucleotide right here. And again, we have carbon 1, 2, 3, 4, and 5. Okay, so this is the 5' end of the bottom strand. And it goes to the 3' end over here. And actually, I did not write in the 3' carbon. So we'll write it in right there. There's carbon number 3. And so DNA is anti-parallel because the only way we can have these hydrogen bonds form, these hydrogen bonds right here, which would be 2 hydrogen bonds holding A and T together, or 3 hydrogen bonds holding C and G together, is because the two nucleotides must be oriented anti-parallel to each other so that the nitrogenous bases can actually form hydrogen bonds. So that's why DNA is anti-parallel, 5' to 3' on one side, and then the opposite direction for the bottom strand, so anti-parallel. Now, when we're extending DNA, you would add a new nucleotide right in this zone on the top strand or on this zone on the bottom strand, because you have to add to the 3' end to continue 5' to 3' extension, 5' to 3' extension. And it's not some magical reason why it does that. It has to do with the functionality of the enzymes that help with DNA replication. So that's our quick review of the structure of DNA individual nucleotides, and then the basic structure of a DNA double helix. Of course, we're not showing the twisting helix. That's not possible for me to draw. We'll save that for true artists. So now let's get down to the nitty-gritty. How do we replicate DNA? So during the S phase of interphase, DNA gets copied. Now in eukaryotic chromosomes, we need to have multiple origins of replication to efficiently copy the DNA found in each chromosome. Prokaryotes is a different story. They have many fewer origins of replications. And an origin of replication, okay, is just the beginning place. So you have a DNA double helix. Where do we begin opening up the DNA double helix, peeling it apart, and it forms a replication bubble. Okay, and again, we're dealing 5' to 3' on the top end. That's the convention of how we write it. And down here, we've got in the opposite direction. And so when we add a new strand of DNA, you would be adding 5' to 3' in that direction for the new top strand, and 5' to 3' in that direction for the new bottom strand in semi-conservative replication, followed by little tiny bits of DNA being replicated following behind the leading strand, which we call Okazaki fragments or lagging strands. But this process is quite complicated, so that means we need to bring in a whole bunch of enzymes and some RNA primers. So what are some of our enzymes? We have helicase, We have single-stranded binding proteins, which technically are not enzymes, but they're important. We have topoisomerase. Oh, go away. And gyrase, though I'm not going to bother trying to draw a gyrase. We'll just use a simple oval to represent them. We have primase to build RNA primers. We have our RNA primers that we're just going to use little T-shaped nucleotide representations, where the top of the T would be the sugar phosphate and the standing part of the T would be the nitrogenous base. We have DNA polymerase 3, we have DNA polymerase 1, and we have ligase. Those are going to be our main players. And we're going to break this down into a 4-step process. Not all of the names that I will use are technical names for each step in each process, but they are descriptive names that help us understand what is happening. So let's start off with the beginning. Initiation. Initiation. So at the origin of replication, so we're only going to focus on one origin of replication. So one opening of the DNA double helix to begin DNA replication. So who are our players? We've got helicase, which is going to break the hydrogen bonds between DNA's two strands. We've got single-stranded binding proteins, which keep the DNA's two strands from retwisting together. And we've got topoisomerase and gyrase, which relieves the stress of the DNA's overcoiling in front of the replication fork. So when you split apart a DNA double helix by breaking the hydrogen bonds, you're opening up a twist. But that causes the areas in front of that opening to supertwist or overtwist or overcoil. How can that be alleviated? Well, topoisomerase and gyrase will break the sugar phosphate backbone, the chemical bonds, the covalent bonds that hold it together, and then it allows it to untwist or swivel and then rejoins back together the sugar phosphate backbone, because you don't want to leave your backbone in pieces, because then the DNA would fall apart and that would be a bit of a problem. So again, this is initiation. So let's take a look at DNA initiation at the origin of replication. So basic rules are as follows. All enzymes read DNA 3' to 5', but they build or write DNA, as it's sometimes put, 5' to 3'. And that's the key for us to remember, because when we are modeling the replication of DNA by our drawings, we are going to be focusing on drawing 5' to 3'. And all enzymes, so all polymerases is what we're talking about. So DNA polymerases and RNA polymerases, which we will talk about in chapter 17. And then, of course, our primases, which will make our RNA primers to get the whole process started. Okay, so here we go. We've got a helicase down here. So here's our helicase, our nice little triangle shape. And helicase acts like a wedge. Functionally, that breaks the hydrogen bonds between the two DNA strands. So here we see our hydrogen bonds represented by these little tiny dots, because they're weaker bonds than covalent bonds. So that's why we always use dots. And so here we see helicase opening up the replication fork. Okay, helicase opening up the replication fork. Okay. And so we are getting a replication bubble drawn. And again, you can see our 5' to 3' on the top, and then anti-parallel on the bottom strand. So now that we've got the basics drawn, and I can pause it here while you catch up with drawing if necessary, but I'm going to keep talking on the video so that if you're reviewing, you don't have to wait and wait and wait and wait. So back to the fundamentals, let's add in the other key players in initiation of DNA replication. So our key players are, we need to have some orange in here, because I want a pen, not a highlighter, and I want orange. So let's draw our topoisomerase. So as the DNA double helix is being opened up here by the helicase enzymes, we're causing an overcoiling, and so topoisomerase, T-O-P-O-I-S-O-M-E-R-A-S-E, and gyrase, okay, alleviate the oil overcoiling and alleviate the stress of the overcoiling by breaking the sugar phosphate backbone in the area in front of the replication fork. Okay, so they relieve overcoiling. You can put more detail in than I can. You have a lot more space to write than I do. Okay, now another thing is you just have to think about the fundamental physics of what's going on here. And some of the fundamental physics tells you that if you open up a DNA double strand, but you have A's near T's and C's near G's, they are going to more than likely come close enough together to try and hydrogen bond again. So how do we keep that from happening? So there are other key players in this initiation process that are single-stranded binding proteins. And there's not any specific number we need to draw in this picture. We just need to draw enough to represent on both sides. Okay, and so these are our single-stranded binding proteins. And they prevent binding, if you want to be fancy about it, it's called re-annealing or sticking together of the double helix. So they stabilize the single strands of DNA and keep them from re-sticking together, so that you actually have an open space in which to work for the enzymes to work and build the two new strands of DNA. So that is DNA initiation. So now we're ready to move on to our next step, which is priming DNA replication, because unlike what you learned in earlier years, DNA replication can't just do its little thing and make A with T, C with G with semi-conservative replication. It doesn't work that way. And then also anti-parallel elongation, because it's kind of one of those, you do this, you do that. You go back and do this, you do that. You do this, you do that. Okay. So we got to prime and then elongate. And then once we open up some more of the helix, prime and elongate. And then you open up some more of the helix by continuing and continuing to open up the replication bubble. Prime and elongate, prime and elongate. But elongation is anti-parallel, leading to the formation of leading strands and lagging strands, which we will talk about. So we have our primase, which binds to single-stranded DNA and reads it from 3' to 5', but more importantly, writes it or builds it 5' to 3'. And not more importantly in nature, but more important to you remembering when you're drawing this. And then once the RNA primer is built, DNA polymerase 3 comes along, yes, Roman numeral 3, and it continues the complementary base pairing, building the new DNA strand 5' to 3'. So DNA polymerase cannot work on a naked single strand of DNA. It needs something to bind onto to help it. And what is it binds onto? It binds onto a short primed sequence, which is 5 to 10 nucleotides long, but it's not DNA nucleotides, it's RNA nucleotides. But that's how we prime in nature. So for simplicity, we are going to eliminate the helicase and the topoisomerase from this image. Okay, and you'll need to draw redraw and make sure that you have enough free nucleotides to make the image clear. So it would be a good idea to go in and this picture doesn't quite have enough free nucleotides in it. So we can imagine that we've just opened up the replication bubble a little bit more. Okay. I'm not trying to draw any particular number. I'm just trying to make more so it'll match my next image that I've got coming up. And it will make it much clearer when we are doing this. So here we go. Next slide. So here we have our primase, which primes the formation of the new DNA strand. And that is going to be priming it using RNA nucleotides. So free RNA nucleotides, actually technically trinucleotides, are going to come in and form a 5 to 10 nucleotide primer. Okay, so this is going to be 5 to 10 nucleotides long. And the primase binds randomly. So it just binds randomly in the open replication bubble to the single-stranded DNA, and it reads what DNA is in the single strand. So if there is an A, it's going to add a U nucleotide. If there's a C, then it'll add a G. If there's a G, it'll add a C, etc., 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 etc. So let's say there's a T here. What's it going to add? It's going to add an A. And so please notice again that primase follows the same fundamental directions we've talked about before, which is 5' to 3'. So 5' carbon to 3' carbon, that direction. So DNA elongation continues that way. And then that means the new strand on the bottom would have to be going in the opposite direction or anti-parallel, 5' to 3' again. And we call this the leading strand because it's going to be, if we take a look here, it's going to be chasing the replication fork as the replication fork continues to move forward as the helicase continues to break the hydrogen bonds and expand. But I'm going to erase those because they will just make our image too complicated. Okay. Then we have behind it, okay, more priming going on. And again, it's 5' to 3'. But this is going to be a lagging strand because it is not going to be following the helicase's replication fork opening. It's going to bump into eventually right here, that primer. Okay, and this one is also another lagging strand. Okay, which I would not suggest using LS to abbreviate because that could be leading strand. Because it's going to eventually, when replication continues, bump into that strand right there when this happens. So we also have another name for that, which you could actually use in an abbreviation, and they're called, it will form Okazaki fragments ultimately, but that's for the next slide when we actually add some DNA nucleotides on. So let's continue to the next slide. Okay, DNA polymerase 3 comes into the picture. And for DNA polymerase 3, we're using this orange square. Now, we are going to pretend that it has started here and continued in that direction. So I'm actually not going to draw it right there. I'm going to draw it a little bit further down. I'm going to draw it down here. And I'll draw it right here. And I'll draw it right here. And I'll draw it right here. So it's already done some action. So what is its action that it's doing? It's adding on new nucleotides. So let me grab my pen. So when it was at the very beginning of the primer, DNA polymerase 3 added on a DNA nucleotide according to base pairing rules, and then another DNA nucleotide according to base pairing rules, and another DNA nucleotide, and another DNA nucleotide, and another DNA nucleotide. And it just keeps going, again, continuously, 5' to 3'. So we're 5' to 3'. So this is our leading strand. Now this strand up here, DNA polymerase, again, connected to the primer, added a nucleotide, 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 and ran into the primer in front of it, and fell off. Okay, so you end up with a little tiny piece of DNA. So we do not call that a leading strand. We call that a, I'm going to change to black, which is easier to see, a lagging strand. And again, it's still continuing 5' to 3'. Everything continues 5' to 3'. It's just the way the enzymes function. They can add a new nucleotide onto the 3' hydroxyl group and therefore continue extending from the 5' end toward the 3' extending end. That's just the way the enzymes do their job. And that lagging strand we can give a name. Lagging strands actually have my absolute favorite science name ever. Okay, they are called Okazaki fragments. I'll give you 3 guesses and the first 2 don't count. What was the name of the scientist who first described Okazaki fragments? Okay, and remember, this is not all happening in discrete. First the one that I drew happened, then the next one that I drew happened, then the next one I drew happened. It's all continuous. So we still need to deal with the bottom piece of DNA down here. So here we see right here, that's the DNA polymerase 3. And so what did it do? It bound onto the primer and elongated. On the 3' hydroxyl group, it added a new nucleotide. 3' hydroxyl group of the new DNA added a new nucleotide. Continuing extension, extension, extension. Okay. But this DNA polymerase is not going to fall off because it's just going to keep going and going and going, following the opening created as the replication bubble expands as helicase continues to break hydrogen bonds and push the replication fork forward. So again, this is a leading strand. So you notice the top and the bottom strands have leading strands on opposite ends of the replication bubble. Okay, and so we just have one final one to deal with. So here we have our primer, add a new nucleotide with DNA nucleotide, add a new DNA nucleotide. So again, to the 3' hydroxyl group, you can add a new nucleotide, DNA polymerase can, and so it just keeps going and going and going. Okay, but then it hits the primer in front of it, it can't add anything, and so DNA polymerase 3 falls off. And I just noticed I drew those really in the wrong color orange, so let me just go over those so that they're in a better color orange to see. Okay. So again, this is an Okazaki fragment, okay, or a lagging strand. So there we see priming and antiparallel elongation. So that's again 5' to 3'. I know I'm hitting this over and over and over again, but you just have to get it. Okay, so we have a gap right here. And we have a gap right here because Okazaki fragment DNA polymerase cannot bind to the primer in front of it. It just can't do that. So what happens next? So let's go ahead. Oh, sorry about that, mic. Or phone, that is. I just hit the microphone with my hand. So I've got some extra stuff in here that we need to get rid of. So for our next drawing, this is not priming. I obviously did not cross this out. So this is step 3. Oh, sorry. Let's see, what do we have here? My pretty pictures, more pretty pictures. Oh, that's just the final. So we see the DNA polymerase is leaving. Okay, so that's wrapping up priming and antiparallel elongation. I just did a lot of drawing on one slide. That's okay. So now let's go to the last 2 steps, which are the basic steps of replacing the primer, because we need to get rid of the RNA nucleotides, and then closing the gap so that we can join together the leading strands and Okazaki fragments, and then Okazaki fragments with other Okazaki fragments, so we can make continuous sugar phosphate backbones within each polynucleotide strand, so that we can actually fully form 2 new DNA double helices, each with 1 old strand and 1 new strand. So replacing the primer, we've got DNA polymerase 1, and it replaces the RNA primers. And how does it do that? DNA polymerase 1 binds onto the 3' end of the Okazaki fragment, and it continues to extend 5' to 3' until it replaces all the RNA nucleotides with DNA nucleotides. But DNA polymerase cannot, and that's DNA polymerase 1, cannot close the gap between the 2 adjacent DNA nucleotides. That takes a totally different player, and that's in the unofficially named step, close the gap, which is where DNA ligase comes into play, and it's going to bind the 2 new nucleotide DNA fragments together. I always like to think of DNA ligase as kind of the glue step. Okay, so here we go. So I'm going to just grab just some sort of blue color to do our, no, I want a pen, not a highlighter. So we're going to do dark blue. So this is going to be DNA polymerase 1 to replace the primer. So what does it do? DNA polymerase 1 binds on here to the 3' end of a new strand. So that's an Okazaki fragment right there. And there's a gap there. And it can actually add a new nucleotide, and nucleotides are drawing in this dark orange. So now it adds a new nucleotide and kicks out that old RNA nucleotide. And then it extends along, and we're going to pretend that it's moving along, and it kicks out that nucleotide. So out goes the RNA nucleotide, in comes the DNA nucleotide. Out goes the RNA nucleotide, in goes the DNA nucleotide. Out goes the RNA nucleotide, in goes the DNA nucleotide. Out goes the RNA nucleotide, in goes the DNA nucleotide. But the catch is it cannot actually bind the DNA that is now adding in to the DNA that's already there that was previously attached to the primer. It's just a weird thing, and it has to do with organic chemistry, and we're not going to go dig into exactly why that is. But so now we have a gap again, but the gap has moved, and the gap is now between where the new DNA nucleotide is that was the last nucleotide of the primer and the first DNA nucleotide that had been added in after the initial primer had been there. Think about it. It's kind of weird, but that's the way it is, and nature is just the way it is. And so now we have a gap. So, oh, I need a black pen. So we have a gap. Okay, and let's go over this one more time. So let me just grab my blue pen again, but this time I'll just go ahead and I'll draw the DNA polymerase further down the road, right here. Let's draw it further down the road because it had started there and gets to that point. So now let me grab my orange pen and go over this again. So again, the DNA polymerase 1 can grab on to the 3' end where there's a hydroxyl group of the Okazaki fragment, and it can add a new nucleotide on using dehydration synthesis. And so it kicks out the old RNA nucleotide from the primer of the strand in front of it and replaces it with the appropriate DNA nucleotide. And then it does it again. Remember, primers are usually 5 to 10 nucleotides long. And then it does it again. So out goes the RNA nucleotide, in comes the DNA nucleotide. Out goes the RNA nucleotide, in goes the DNA nucleotide. Out goes the RNA nucleotide, in goes the DNA nucleotide. But in that process, you break the covalent bond that held what had been the RNA primer to the actual DNA strand that had been elongated off of the RNA primer. And so you've replaced the primer, but it is now actually covalently part of the Okazaki fragment that was behind it. Yep, behind it. It's strange. It's nature. It is what it is. Okay, so now we have a gap. Okay, and again, this is 5', this is 3', this is 5', this is 3'. Okay, notice we still have these primers at the front end, as it were, the 5' end of the Okazaki fragment. Those did not get kicked out. It will have to wait until you further open up the replication bubble, and you can throw DNA polymerase in on the strand behind this Okazaki fragment to replace that primer. Okay, so we've got gaps. Oh, did I forget to erase that, or did it just not erase very well? So there's our gap. Okay. So how do we close the gap? Along comes DNA ligase, or the gluing enzyme, as I like to think it. And we're just going to use a simple little cross here. Don't know who came up with the symbol. Somebody did it in some notes that I saw once upon a time, and so it's just what I use. Hey, as long as you have a key and it tells you what it is, that's good enough for me. Hey, that was pretty funny. It just magically made a strand on here. So now we bind that strand, and down here we bind this strand. And so now we have one continuous sugar phosphate backbone of DNA nucleotides, one continuous sugar phosphate backbone of DNA nucleotides. Okay, and we still have our primer here. If you can't see it, we still have the primer here. See that? That's a primer. We still have our primer here, RNA primer here. Let me just take those away. Come on, go away. Okay, but those will have to wait until we further open the replication bubble, and then we can get rid of those primers by again following the same process. So DNA ligase comes in, connects the former Okazaki fragment to the leading strand, and now it is one unified strand. Okay, here's the Okazaki fragment that was, here's the leading strand. They are no longer Okazaki fragments, and it just is now part of the leading strand that is going that way. No longer Okazaki fragment, it is now part of the leading strand that is going that way. Until we form more Okazaki fragments as we continue. And that is the process of DNA replication when you just focus in on one replication bubble starting at one origin of replication. And again, going back to, let's look at our main players. Helicase separates the DNA double helix. Single-stranded binding proteins stabilize the single strands. Topoisomerase and gyrase alleviate the over-twisting in front of the replication forks. Primase adds 5 to 10 nucleotides randomly to make an RNA primer, which can then allow the DNA polymerase 3 to follow right in front of it and continue extension and make DNA nucleotides join together by dehydration synthesis and do DNA elongation. And then DNA polymerase 1 comes along and removes the RNA primers and replaces them with DNA nucleotides. And then finally, to connect the sugar phosphate backbone of the new strand of DNA, we rely on ligase. And that is DNA replication in short, or not so short.